Hi everyone, we're going over chapter 20, the blood. So, the major components of blood. So, blood is used to transport nutrients, oxygen, carbon dioxide, waste, hormones, and immune system. And this maintains homeostasis for the interstitial fluid. This fluid is the extracellular fluid, and this distributes heat throughout the body. So blood travels through the circulatory system and it's made up of your heart and blood vessels. So this is actually a connective tissue because there's cells suspended in the matrix. So in the blood it's made up of many different blood cells and fragments. And the matrix is actually plasma and is the fluid portion of the blood. Okay. And it is made up of different parts of like multiple parts. So it's made up of matrix and cells. And the plasma is composed of 55% of blood volume and 92% of matrix is water. And the proteins make about like 7% of it. So you can see, so 45% are these form elements, which are like the little cells. The plasma is made up of multiple different other parts, including water and proteins. So as you can see, there is a lot of neutrophils, a lot of different types of white blood cells, but it only makes up about 0.1%. So if you look at a blood that has a little bit too many white blood cells, you might ask, like, what is going on? So red blood cells are actually in flat biconcave discs. Biconcave means you see this little what is it called? This little cave right here? That's biconcave. And this would transport gases throughout the body. So this shape actually increases surface area and which allows for faster gas diffusion in and out of the red blood cells. So it also transport oxygen, carbon dioxide, and nitrogen. And this has a very short lifespan because there's no nucleus, there's no mitochondria, ER, Golgi, vesicles, and etc. So what happens? So red blood cells, the way that they are created is they start up in the red marrow with all these organelles, but as they mature and then develop into mature red blood cells, they need to make room for hemoglobin proteins. So then they start getting rid of these organelles. So, okay, let me break it down for you guys. So, what happens is, so bone marrow, right? Let me draw it out for you guys. Bone marrow right here. And then all these little guys, all these little red blood cells come out like wee, wee. Okay. And what happens is they, as they mature, become these guys. They get rid of everything. They're like, bye nucleus, bye mitochondria, like bye to all those parts. Reason why is because red blood cells carry oxygen and they need space for the hemoglobin. So they get rid of all of those so that they can, do their main job, carry oxygen. Because, all these little parts, they take up space. Okay, let me clear it now. Okay, let's move on. Hemoglobin. What is this? So these are the oxygen transport proteins in the red blood cells. Remember how we just said red blood cells carry oxygen? Well, they get rid of all their parts to have hemoglobin. Hemoglobin is majority of the inferior and majority of volume of the cytoplasm of red blood cells. There are about 270 million hemoglobin molecules per red blood cell. It fills up about 1/3 of each red blood cell. And what they have is they have four iron groups called the hemigroups. And the hemigroups bind to oxygen. Every hemoglobin protein or molecule carries four oxygen at a time. So think of that. 4 times 270, that's like 1 billion. So it carries a lot of oxygen. And when it binds to oxygen, it turns bright red because of the iron molecules that you have in the hemigroup. It's kind of similar to like rusted iron because iron binds to oxygen. And what does it do? You know that little like when you put nails exposed to air for a while, it turns a little rusty. And without oxygen, hemoglobin actually turns to a darker gray color. And it's seen in blood. Blood without oxygen are deeper, darker red color, not blue, but it's just darker red. So what happens is The hemoglobin, right? It has these four hemigroups: B chain, A chain, like okay? And each one of these carries four oxygens. So then, and there's so many of them, so many hemoglobins in one red blood cell. So red blood cells carry a lot of oxygen. Okay, they're big, big. Yeah. Iron binds to oxygen, turns red, and without oxygen is dark gray. Dark gray like this. This is no oxygen. This is oxygen, bright red color. Let's go through the red blood cell cycle. They have very short lifespan. They only live there for 120 days and then they just die. They're just disappear. Okay, so let's start with the first step. The first step is erythropoiesis. Erythropoiesis is the production of red blood cell. Okay, so all red blood cells are produced in the bone marrow, and immature red blood cells mature and enter into the bloodstream. The hormones involved are erythropoietin from the kidneys, and this stimulates red blood production. And they leave. Okay, so see right here? This is erythropoiesis. Leave the bone marrow and they circulate doing their job for about 4 months. They're like, okay, I'm gonna do my job. Like, I'm employed 4 months. And they get old and they get less efficient. Why is this? They have no organelles. So, what they would do is to replace their proteins and repair they can't replace their proteins, so they get rigid. And one day, as they're traveling through some of the organelles, like the spleen, liver, back through the bone marrow, the macrophages notice the red old blood cell isn't functioning properly. Let me see if there's a step for that. They're like, hey, I see you're getting a little old. You're looking kind of rigid. So macrophages are like, I'm gonna do phagocytosis. And they just gobble it up. Like they eat it off. And the red blood cells then die, and the macrophages dismantle all the hemoglobin proteins so that the body can recycle them. And the globin, which is the protein, part of the hemoglobin, gets broken down and used by the whole body. Hemigroups that contain iron also gets broken down. Iron gets carried to the liver and sent back to the bone marrow. So what happens is phagocytosis happens, and they're like, okay, let's reuse, like recycle. So they recycle the protein. They also recycle the iron. It gets carried to the liver and sent to the bone marrow to make new red blood cells. The rest of the hemoglobin or hemigroups get broken down to a protein called biviridin. Biviridin right here. Okay? And and then it goes to the liver and it's okay, so biviridin is green and bilirubin is yellow. And the liver processes it and puts it in your small intestines where it joins with the rest of your feces and gets secreted out of your body. So it goes through this step. And the major components of blood. So let's start with leukocytes. So the production of leukocytes is called leukopoiesis. So other one is erythropoiesis, leukopoiesis. And they are big round white blood cells. They are much larger than the red blood cells, and they act as an immune cell against pathogens like bacteria, viruses, worms. And their modes of defense is phagocytosis, where they engulf it, they form a vesicle, and then they break it down, or they produce chemicals or antibodies that attack the pathogens like toxins that would kill the worm. So they would secrete chemicals, or they have antibodies or chemicals so they attach to the pathogens and target immune systems. So the immune system tracks them down and destroys that bacteria or viruses. Leukocyte diapedesis. Dia- Diapetes. Okay, so this is when the white blood cells leave the intact blood vessels by squeezing between cells, and they wander through the body's tissues to fight off pathogens. So they travel through a capillary and decide, oh, there might be some pathogens. So it squeezes through the walls of the capillaries, exit the capillary, and wander through connective tissue to find the bacteria. So this is immigration. They're like, okay, start coming out, coming out, coming out. And they're like, okay, let's go fight some pathogens, you know? They're like warriors. Warriors are like, okay, like, who do we get rid of now? There's multiple types. Mhm. So, first one is the neutrophil. The neutrophil essentially, don't make noise. I'm making a video for class. Bye. Okay, so let's go over each of these. Neutrophils are the most common. There's about 54% to 60% of leukocytes. Also for I want to tell you guys about the red blood cell cycle. I wanted to add some more things. So first step is production, second is circulation, third is recycling, fourth is excretion. Liver processes bilirubin and secretes into small intestine, thus bile. And this is how you remember it. Big red soldiers live brave and move boldly and march home boldly. What does that stand for? B as in, okay, B as in bone marrow, R as in release, S as in aging, L as in liver, spleen, bone marrow, M as in iron, H as in heme, and B as in feces, which is basically all the steps. So I wanted to give you guys like a small tip. It's a little hard to remember it, so I have a few techniques on how do I do this. So what I do is, for example, for neutrophil, right? I notice this phagocytosis, they release enzymes and they do an extracellular trap. They die after battle though. They're suicide mission basically. If there's a high count of neutrophil, you know you have an infection. Because you remember how I said they release warriors? If they're releasing too many neutrophils, that's mean you have something going on that's not right. And I call these the ninja eaters. Eosinophil, I call these the exterminators. Why? Because the way that they attack is the chemical release onto the parasite in worms. So, exterminators, what do they use? They use chemicals to kill bugs, right? Eosophil is the same. Eosophil exterminators, they attack chemicals, they, they use chemicals to release onto parasite in worms. If you have a high count of these, it means you have a parasite infection or allergies. Why allergies? Because allergies, your body is fighting. Your immune system is fighting against that invader. Like, let's say you're allergic to dogs. It's fighting against that. And basophil. Basophil, blast the alarm. So this one secretes heparin, histamine, serotonin, and it triggers inflammation and allergic reaction. Okay, so they secrete chemicals and they like the chemicals I told you guys, and basically it's telling you like you have a symptoms of allergic attack. So your eyes start to water, your mucous membrane is swollen, your nose gets stuffy overall. You know, blast the alarm, like, oh, you have allergic reaction, your body's telling you, help, help. Monocytes. Monocytes is mega cleaners. This is funny. So they attack with phagocytosis. They leave the bloodstream via diapedesis, become macrophages, and they destroy bacterial viruses, clean dead cells. They then fuse into osteoblasts to break down old bones. They're like the cleaners. They're like, hey, I'm gonna clean this up. I'm gonna leave my place clean. Okay. And then let's go through. So they only do phagocytosis. and they destroy bacteria. Yeah, and mer okay, and then let's go over lymphocytes. So these two are granulocytes, which means you can see the grain, while this is agranulocytes, which means you can't see the grain. Lymphocytes are small rounded nuclei. They are T cells or T lymphocytes, B cells, and all three of these do different modes of attack. I call these the laser specialists. Okay? So T cells kill infected cells, B cells produce antibodies, and NK, which is a natural killer, attack viruses, infected, or cancer cells. So you have a lot of natural killers, that means you have cancer. Okay, so with a leukemia patient, remember how I said, told you guys, they release warriors? If they release too many of these leukocytes, okay, that means there's an infection going on. With Esophila, what does Esophila do? Remember what I told you guys? They're the exterminators. If they, and then if you need a lot of exterminators if you have a lot of termites. So same thing. You have a lot of parasitic worms, you need a lot of exterminators. Neutrophila, bacterial infection. Lymphocytos, cancer. Remember lymphocytos? Not they're laser specialists. Neutrophila, they're ninja killers. Platelets. Platelets are those little they aid in blood clotting. Co co coag coagulation. And what they do is they stop the bleedings from about like they are tiny little flattened disc cell shaped. Okay, so they're like this cell and they become they start up as megakaryocytes. Mm, so yeah, megakaryocytes, and then they start to break up into fragments. And the platelets contain a small packets of cytoplasm inside a complete membrane. Kind of similar to red blood cell, they have no organelles, but they live only for 10 days until they get gobbled up by the macrophages. Macrophages hang out in the spleen and liver, and they're like, oh, you're getting old, and they just like, yep. Platelets do have mitochondria though, and fragments of ER, but they have contain vesicles that contain clotting factors to help with blood clot. Okay, let's go over the blood clotting. So first, what happens is, okay, let me explain it to you guys and break it up. So basically, um, they will release clotting factors and trigger the fibrinogen to unfold the cells and then form this net that would trap the red blood cells. And it's sticky, so the platelets will stick to it, and this will stop you from bleeding out. So it triggers homeostasis. So these platelets, right? They see a damaged vessel. You know, like let's say you get a cut, you cut, and then what happens? They're like the platelets like, okay, let's go, let's go, let's go, and then they start releasing these clotting factors. They're like, hold up, let me draw it out, guys. Let me draw it out. They're like, okay, clotting factors, come out. Okay, that's why I imagine it like, and this would trigger fibrinogen, right? So fibrinogen unwraps and it forms a sticky net. It's like, and then it traps the red blood cells. So, because you know, when you cut, blood rushes over and the platelets come. platelets release the fibrinogen. Fibrinogen makes it unfolds itself. It makes it sticky. That's why you have like little clots, and this stops you from bleeding out because you don't want to. Let's say get cut, right, and you keep bleeding, bleeding, bleeding. What happens? You just run out of blood and you just die. So that's why this is very useful. Okay, and the hemostasis, it's like when you have a rough surface inside your body, it causes unwanted blood clots, right? So this is a nice clean surface, an area where you have rough, it's ugly, it's not straight. Your platelets are like, okay, like let's form. Forms a huge blood clot because this is why strokes happen or tissue death because blood can't flow to that area. So you just lose the tissue. Okay, and then let's go to homeoposis, the development of formed. Okay, so how you form your blood cells is you start with the pluripotent stem cells. These stem cells divide into many different types of cells, and they go through a long process where each one of them is stimulated by different hormones to create different types of blood cells. For example, erythropoiesis, red blood cell. This is triggered by the hormone called erythropoietin, like what we said earlier. And erythropoietin is produced by the kidney that promotes erythropoiesis. And it tells the bone marrow, hey, go to erythropoiesis. And this is made by a negative feedback loop. You know, like beautiful too. And you know how I said red blood cells carry oxygen? So when you have low blood oxygen, it triggers erythropoietin secretion. And so what happens? Let me break it down, guys. So the kidneys are like, oh my God, this person has low oxygen. And then they're like, okay, let's go make some red blood cells. So erythropoietin's okay. They're like, okay, I'm on it. So they start releasing. Let me get my markers. So you're like low. You're like, oh my gosh, like running out of oxygen, right? And then the kidney's like, hey, oop, hey, let me get you some. That's what I imagine it like. Let me get you some. And then afterwards, erythropoietin's like, I got you. Like that. And then afterwards, red blood cells get released and they're like erythropoietin, this is like the precursor, right? And then this starts creating more red blood cells. So red blood cells are like released. They're like coming, coming. They're like, I'm coming. And then afterwards, this will raise your oxygen level. Why? Oxygen carry red blood cells carry oxygen. Okay? And and then when your kidney it sees that oxygen is normal, they're like, hey, oxygen is normal now, right? We don't need no more. And then what happens is they're like, we're stopping this. You don't need any more. I don't need any more. Let's stop erythropoietin. And then it's done. So summary? Let me summary. So what happens? Kidney's like, let me get some. And they're like, erythropoietin's like, I got you. Let me get some red blood cells. Red blood cells like, okay, let me get some oxygen. And then liver kidney's like, I see you got enough. And then they just stop. That's negative feedback loop in like yeah. So that's it. Okay, so hemocoagulation. is you have antigens that are receptor proteins or glycoproteins on the surface of your cells. Antibodies are plasma proteins. They are target anything that doesn't belong in your body. So if you transfuse, like let's say someone's type A positive, and you give them a B positive, what's gonna happen? It's gonna hemocoagulate. It's gonna start attacking and then form this huge clot. So antigen is the glycoprotein, antibody is the opposite. So this is how I like to think of it. Group A has A flags. It doesn't have a B flag, so it will attack the B flag. I think they're constantly at war. I like to think of war. Attack and then yeah. Antibodies travel through the blood plasma and attack any and anytime it finds an antigen that doesn't belong in your body, it will attack it and destroy it. But if you give a foreign one, it will ignore the body's native origin. It starts doing this whole mess. Yeah. So basically, with blood typing cards, in order to figure this one out, it's pretty simple. So I look at anti-A, I look at the anti-D. Anti-D means RH, RH means positive or negative. This person has clumping on A and D, A positive. Yeah. And that's why it's coagulating. Why? You drop the blood in, it starts clumping because they're like, hey, buddy, buddy. And then, yeah. Let's go to the hemolytic disease of the newborn. This is when a Rh negative female becomes pregnant with a Rh positive fetus. And it's not too dangerous in the first pregnancy unless the female has sensitized to Rh positive blood. Like, let's say she shared needles, she had the wrong transfusion. So, the placenta can actually prevent the baby's blood from traveling inside the placenta. It's protected, the baby is like warm and safe here, they're not mixing blood. Okay, let me draw this out. So, this mommy is Rh negative. You don't want to mix those two. This baby is Rh positive. Okay. That's a very ugly plus sign. And the first one, they're like, okay, like I'm safe, I'm safe. Like I don't need to, the placenta will protect me. But the placenta can prevent, yeah, the placenta can prevent the baby's blood from traveling across the placenta, and her body won't be creating Rh antibodies until it sees the little bits of the baby's blood cross the placenta. But during the delivery, it is common for the placenta to rupture. So, like deliveries, there are things happening. Sometimes your placenta ruptures and it will leak. So the baby's blood might escape to the mother's bloodstream. If that happens, her body will become sensitive and produce antibodies that can attack the Rh antigen on the Rh blood. So the blood leaks, right? Rh positive blood leaks to the mom. And then it starts being weird. It's like the Rh negative's like, what are you doing here? Like, and Rh positive like, leak. Did he like, what am I doing here? And then so this can actually become an issue if it has a second child and the child's Rh positive, because if she has Rh antibodies that can cross the placenta, bind and attack the blood of the developing fetus, and the baby will suffer death or severe diseases like enlarged heart, spleen, or liver. And there's actually a drug called RHOGAM, and this would bind to the Rh antibodies and prevent it from attacking the fetus. So they're like biting. Okay, they're like, don't attack the baby. Don't attack the baby. Protect, protect. So these are like the protectors. And then the baby's like RH positive, right? He's like, and even the blood leaks, the antibodies are like, it's okay. The blood is safe. We don't have to attack you. And yeah, this is why prenatal screening is very important. Yeah. And that is all. I hope that made sense.